We are now going to talk about the another lecture in this session, the microbial communities system and quorum sensing. Now, before I go into detail about the microbial communication, let me talk about the basic introduction about this topic. As we discussed in detail in my last few lectures and many lectures about the microbial world and the varieties and species of microorganisms of different class, different category, different groups, and they have a different function as well. Some are useful, beneficial, harmful. So how these microbe and microbial world communicate each other. For example, we communicate through speaking and we think through our brain. But the question, do these microbial world or do these microorganisms have brain or tongue to speak, to communicate through what they are thinking? No. But they have some signal process, and this signal process is controlled by certain metabolic processes, metabolic reactions, which is carried by certain genes, right, or proteins which are coded by certain genes on the genome. And this system is called quorum sensing system. Why we call it quorum sensing system? We call it quorum sensing system because they make a complete quorum. Until unless the whole quorum is formed, the senses will not move further, right? So we talk about the quorum sensing, then we talk about the history of quorum sensing, mechanism, and then significance of quorum sensing. Now, what is the quorum sensing? Quorum sensing is a bacterial communication system or communication mechanism that uses the chemical signals which are produced by several proteins, enzymes, reactions in the cell and coded by certain genes, and these are called autoinducers to regulate the gene expression. The autoinducers allow the bacteria to coordinate colony-wide behavior such as a sporulation, bioluminescence, virulence, conjugation, competence, and biofilm formations. So all these action which is controlled by the inducers, autoinducers or quorum sensing genes are showing the result, are showing the phenotypic expression in these forms, and that reflects that quorum sensing mechanism is working. If we block this quorum sensing mechanism somehow, then we can lose conjugation, we can lose competence, we can lose several virulence factors and other actions carried out by the bacterial system. Now, as far as the history of quorum sensing is concerned, in early 20th century, Streptococcus pneumonia was first observed as a cell-to-cell communication in gram-positive bacteria. The peptide signals which trigger, which enhance, right, or let the DNA uptake, we call the competency of the bacteria will produce, and that make the cells more like competence and having the virulence factors released through these mechanism. That was the first, the indication of the understanding the quorum sensing mechanism. In 1970s and 80s, between in 10 years, Vibrio fischeri is one of the bacterial species, and that was discovered in AHL based quorum sensing mechanism. This is autoinducers, and we call them as a quorum sensing mechanism in gram-negative bacteria, and that is it controls the bioluminescence in marine fish, and it's quite host. So having high and low bioluminescence which can be visualized with the eye and right with the microscope will be controlled by these AHL based quorum sensing mechanism. Later, the several development takes place, several discoveries came into pictures which recognizes the diverse signals like AHL's oligopeptides, AI2, autoinducer 2 involved in the quorum sensing signals, and they basically regulate through promoter, they regulate the expression of several genes which are involved into several virulence vectors, several biofilm formations, or several other quorum sensing genes which help in order to sustain the bacteria. It found in many gram-positive and gram-negative bacteria. Both gram-positive and gram-negative bacteria carries these quorum sensing mechanism which regulate the several virulence factor, symbiosis, and biofilm formation. Now, in this chart, you can see the signals and structure of these. So, N-acetyl, this is the N-acyl homoserine, we call AHL lactone. This AHL is the structure of this aromatic compound with the this is the ring compound, and this compound is used for the various bacteria like Vibrio fischeri and then Agrobacterium tumefaciens, which is one of the important plant-based bacteria carrying several plasmids which helps in the plant transformations and provide the colonies to help in making transgenic plant in natural manner. Pseudomonas aeruginosa, Vibrio, Harvey, and there are several more gram-positive and gram-negative bacteria that carries the HL. Similarly, you have AI2, we call furanosyl borate is another autoinducers, and this is a quorum sensing molecule. Then cyclic thiolactone is another quorum sensing molecule. They are different quorum sensing molecule. They have a different action like bioluminescence. They may also call some virulence factors of the bacteria. Then, for example, cyclic thiolactone AIP2 is a structure. This structure is carried. This structure carried in Staphylococcus aureus, and they are leading to virulence factor. So, inhibiting these AIP2, you can block the virulence factor. So, this is how the importance of this quorum sensing and to understand the quorum sensing mechanism. Similarly, you have several more. You have a methyl dodecanoic acid, then again for virulence factors, and then we have for Candida albicans we have another interesting molecule we call the farnesoic acid. This is again involved in the dimorphic transition and virulence factors. So they promote the growth of these fungus, Candida albicans, which is a class of fungus. Then we have a 3-hydroxy tridecanphoone, it's present in the Vibrio cholera, that is cholera causing bacteria. Again, it enhances the property of the bacteria to cause diarrhea and cause the several symptoms of the cholera. So it is also controlling the virulence factor. Now, the mechanism of quorum sensing is basically several mechanisms are reported having several molecules, because every molecule having different structures have a different mechanism of action, but the molecules are also produced as a byproduct in the cell, which are produced, these chemicals are produced in the cell as a signaling process through several protein, several metabolic pathways, as a result of several metabolic pathways. Now, if you look into individual bacteria produce autoinducers during the growth, the gram-negative bacteria, they produce acyl homoserine lactone. This is autoinducer that diffuse through the thin layer of the cell wall. So this is how the mechanism acts, as it can enter through cell wall, and then while it enters into the gram-negative cell wall, because in gram-negative you have a less peptidoglycan than gram-positive, wherein gram-positive bacteria produce the peptide-based autoinducer, which produces the via ATP binding cassette, because it is a thick membrane, so it requires certain energy to generate through ATP hydrolysis, and they help this peptide based autoinducer help to enter into the bacteria, and these bacteria population increase the extracellular autoinducers concentration rise, eventually reaching to threshold, that is a critical mass where it can cause certain action based on this quorum sensing. Now, one threshold is reached, intracellular autoinducer concentration increase bind to receptor, trigger the signaling cascade and alter gene expression. Now, gene expression is also controlled by the signal molecule. What genes to be expressed under the regulatory mechanism is all controlled by these quorum sensing signals. Most of the time they involve in negative feedback, so it reduces the autoinducer synthesis after activation. So once it activates the action, either virulence or competence or conjugation or any virulence factors through certain mean, then they can stop these through the same mechanism as a reducing the by feedback, we call negative feedback mechanism. So the high expression of these autoinducers, they can again become a negative suppressor molecule, become suppressor to suppress the process of the action, and they can reduce the action if it is not required further in the cell. The overview of how quorum sensing work in bacteria, just have a look at this is the picture showing it was taken by John Windsors, and who has published these figures, and this is called extracellular concentration of this molecule, and this is intracellular. So extracellular are more than intracellular. Now, the cell continue to replicates, and after replication you see the extracellular concentration is equal to the intracellular so extracellular concentration is less than intracellular, but after replication you see the almost equivalent to extracellular concentration and intracellular concentration are almost equal. This we call replicated cell also begin to produce the autoinducer. Now similarly, when intracellular AI stop moving out of the cell, then you can see the replication is stopped, but the signals stop synthesizing and their extracellular concentrations increase of the autoinducers, and then intracellular concentration get reduced. Now again, when high AI concentration, autoinducer concentration down regulated itself by inhibiting its own transcription factor. So when the concentration is increased in outside cell, these outside cells further inhibit. So this is called feedback mechanism, or we call negative feedback. So this overexpress signals, when they are not used and they are more than what they are inside, then they go back and suppress the gene regulation and transcription factors, and by reducing the transcription factor they can down regulate the process of autoinducer synthesis, and this is called feedback. And then again you have extracellular signals are less than the intracellular signals in the later stage. Now discovery in gram-positive bacteria Streptococcus pneumonia first as an autoinducer, and this pneumonia bacteria produce the releases a peptide signal into the environment. Now the population increases, peptide concentration rises, triggers some cells to become competent for cell uptake transformation. So this is how the transformation takes place, and so the large number of molecules are produced. The competent cells release the bacteriocins, and these bacteriocins that lies non-competent cells which are not competent enough to compete with these cells which are producing these quorum sensing cells. Now this release the DNA and virulence factor, and so it can also help in inhibiting the several bacteria. Now this process contribute to pathogenicity, pneumonia and meningitis. So by increasing in number or the threshold number of these Streptococcus pneumonia, they will lead to certain infection. So now inhibiting the virulence factors, or inhibiting the bacteriocin productions, or inhibiting the competency factors, are inhibiting this quorum sensing molecule which are produced by the Streptococcus pneumonia, and the pathways which helps to produce this signal molecule can be breakdown, can be stopped. So that now the new approach is to look into the targets, quorum sensing targets, and these quorum sensing targets can basically inhibit this well and reduce it. Now the species-specific quorum sensing, we call the example Vibrio cholera, use quorum sensing to regulate the biofilm formation and virulence. Low autoinducers level receptors act as a kinases activating LuxO. So these are the gene LuxO genes, LuxP genes, and various other Fimri genes. They promote the biofilm cell expression. Now after the expression of these genes, the biofilm formations may also increase by cell-to-cell interaction. Sometimes these genes also inhibit in certain cases, so it has a dual role. Function may be different, but the structures and remain same, but it can act inhibitor for the other microorganisms. Now high inducers level like receptors switch to phosphatase and dephosphorylating LuxO, and this shutting down the biofilm production. So this is how the mechanism of a transcription, that is the regulation of the LuxO gene. So it can, when the more LuxO gene is produced, biofilm formation, then again the same phosphatase produced with dephosphorylating. This phosphatase is basically removal of the phosphate from this gene, from this protein, from this gene, and then after, when the dephosphorylated state, it can downregulate it. It can suppress the biofilm production. So quorum sensing is a therapeutic target. If you can use the quorum sensing signal, or if you block the quorum sensing signal, artificially increasing the autoinducers can halt biofilm formation. So this is another very interesting area, that to how to identify the quorum sensing signals to use as a target to inhibit it and to block it. Now the quorum sensing system of Vibrio cholera was also discovered in 70s and 80s. It uses the regulate to biofilm formation. Now if you look on the left side panel, that is low cell density. In the low cell density you have low AI, that is autoinducers, and phosphate flow. This phosphate LuxOP get phosphatase, and then in the low cell density concentration, the quorum sensing signals sRNA is formed, and HAPR mRNA is again produced, and this is the responsible for the biofilm formations or the virulence factor. Now this increases it. Now in case of high cell density, when the AI signal very high, autoinducer is very high, and again dephosphatase takes place and the suppression of the LUXO gene takes place, and this is again reduce the biofilm formations in the virulence factor. So basically the regulation of the expression of these virulence factors through quorum sensing gene or quorum sensing molecule is controlled by its own sometimes. So autoinducer is basically can use as a negative feedback to control it at several stages. Now look at this picture. This is a very nice figure. This is the Vibrio species, and this is the low density and high density example. This is autoinducer 1, 2, Cal1 and DPO. So these are some molecules which are involved in quorum sensing molecule. So AI1 is LuxM, LuxX, then CQSA, TDH. So these are all quorum sensing molecule. We call quorum sensing molecule. These quorum sensing molecule, how do they act? And so, for example, in this case, you can see the LuxN, PQ and Q. They are the molecule which are entering into the membrane C, and they are basically activating the phosphorylation of LuxO, LuxU and LuxP, LuxO to call phosphorylation. Now the phosphorylation increase the at low density, increase the activity of this QRR, the quorum sensing molecule, that is RNA molecule, and this is activating the FA and LuxR, LuxP, which basically induce the production of the biofilm formations and other. Now similarly, if you look into the high density, that is when the large number of molecules are present here, see, in this case the dephosphorylation takes place and LuxO will not activate and group behavior is changed. Again it will trigger the action that how it again activate when it is required for the biofilm formation. So this mechanism both in quorum sensing, that is low signals and high signals through this mechanism of Lux U to LuxO is basically involved in the production of the quorum sensing signal. Now in case of Pseudomonas aeruginosa, you have a different molecules, quorum sensing molecules are present. In Streptococcus aureus, you have a different molecules are present. All these molecules are involved in a different interconnected metabolic pathways, where you can have many metabolic pathways are being interconnected through these molecules at low phosphate and high phosphate. Accordingly they can have signals produced. Now so the take home message from this is that how you can make use of these molecules used as a target for inhibition of these quorum sensing molecules, to inhibition of the biofilms, virulence, competencies, conjugations that are these are the responsible for the growth of the cells, multiplications, proliferations, and of course infection in the animals and human. Now interspecies quorum sensing is defined as interspecies quorum sensing defined as the within the species, right? So we have two species, they have signals, they produce each others and give the signals to communicate the signals and what to do, whether you want to be more virulent, infectious, or you want to be less infectious. This is all talk to each other. So they basically this is kind of a cross talk between two different species. So some bacteria cannot produce autoinducer but can respond to the signals from other species. So for example, there are two species. So one species is not producing the autoinducer, but the other species is producing. So when the other species in the communication or in the community, they give the signal, the other bacteria will respond and enabling the environmental sensing. And this sense is also called quorum sensing, and they help either way, whether it can evade that microorganism, they can remove out from the system, or they can also allow them to grow. So it can also enhance the growth in certain condition. It can also suppress the growth in certain condition. It can lead to competitions and cooperations among each other between two species. For example, the bacillus spores in the gut disturb the Staphylococcus aureus AGR system using the fungicin or fungicin reducing virulence factor. So basically this is a good example of bacillus where it inhibit the Staphylococcus aureus, right, AGR system using the fungicin reducing virulence factor. Example of enhanced virulence like Burkholderia, Capacia, Pseudomonas aeruginosa, where we have a co-infection. Co-infections means like both the cells are infected that increase the virulence factor production in via QS crosstalk when the two species are crosstalk each other. So they can enhance the process of quorum sensing and signaling process for the growth of the cell. For example, the cooperation is we have example of Porphyromonas, gingivilis, and then Streptococcus cronii from the mixed species. So sometimes it has been observed that polymicrobial, we call our polymicrobial infection or polymicrobial biofilm formations, where the two species or two microorganisms, even from a different genera. For example, if you talk about the Streptococcus aureus and Pseudomonas aeruginosa, they can come together, and they can also behave like they have different behavior, and they can behave to facilitate the infection establishment in that particular site. And biofilm could be formed, could be disrupted. It all depends on what kind of a signals are being produced by each microorganisms or each species. So polymicrobial infection is also a challenge for the physicians to control. Now look at this fascinating picture where the quorum sensing molecules interaction in algal bacteria system, gram-positive, gram-negative bacteria, and all AIP, auto-inducing peptides, AHL, acyl homoserine lactone. Now this is a well descriptive, like this is the bacteria, so this is the fungus. So this is they are trying to show how the interlink between these two kingdoms. So we call inter-kingdom communications. So interspecies communication is easy to understand, but inter-kingdom communication is also possible. For example, one is from different kingdom, algae and bacteria. They are from two different kingdom, two different world. So this is interspecies interaction, but this is interspecies interaction, but this is interkingdom interaction, where they have shown the quorum sensing signals, they have a crosstalk. So IAA and AIP, they have produced these kind of signals, and these signals are connected to quorum sensing analogs, phospholipid, tryptophan in algae, and the genes are transferred. The genes are transferred through certain means, and they can, after transferring the gene, they can also have extracellular signals molecules can also produce, which can help, which can also trigger the molecule here. So this is a mechanism where it can work in inter-kingdom species, and as a result, you see the mutualism take place. It could have the commensalism, mutualism, as we already discussed in earlier, what the commensalism means and what is parasitism means. So mutualism means like they both are interconnected and they are benefited each other, right? But in other, one is like surviving, the other is not affected. But in parasitism, one is activated, the other is like inactivated. So one is growing and other is like so losing the lives or inhibiting. So in both the case, interspecies or inter-kingdom communications also, with a good example of bacteria and algae, you can have commensalism, mutualism, and parasitism, where you can see how these quorum sensing signals work, and they can either help to other species to survive, or they does not help to survive, and rather it inhibit the other species to survive in the environment. The quorum sensing molecule signals that their functions are this is the molecular signals, N-acetyl homoserine, oligopeptides, autoinducer A2, cyclic thiolactone, and other molecules present. This is the representative organisms: Vibrio, Pseudomonas, then Vibrio, Harvey, and then Streptococcus pneumonia, Streptococcus aureus, subtilis, and so these are all list of the microorganisms which are seen here. These lists of microorganisms are governed by these signal, that is quorum sensing signal, and these are the actions taken by these bacteria, like bioluminescence. Then you have virulence factors. Sometimes you have biofilm formations and plasmid transformation, competency uptake in case of oligopeptide autoinducers, conjugations, then virulence regulations. So all these, this table is important to learn that how this basically help you to understand about the different virulence factors and their expression of these virulence factors as a result of the quorum sensing signal molecule from different bacteria. Now, having this information, these quorum sensing targets can be used as a intervention for the various molecules to inhibit and to control the infection and to suppress the action of these bacteria. Now, the significance of the quorum sensing molecule is is like coordinated gene expression. It can coordinate the gene expression. It can coordinate the gene expression. It enable the bacteria to synchronize behavior at the colony level, even in the liquid cultures, uh, in the biofilm formations, sporulations, and virulence. So how they coordinate each other. Then virulence regulation is another important point where it can regulate the virulence factors. So it in turn also control the pathogenicity of microorganisms, how pathogen the microorganisms. Suppose there are some variants takes place is more pathogenic. So it means there are some genes are responsible for reduction or expression of the pathogenic molecule, virulence factors. So, like Vibrio cholera, Streptococcus aureus use quorum sensing to control the toxin productions and host colonization. So, this is another important function. Then biofilm formation is another thing. So, quorum sensing coordinate the biofilm formation. The biofilm is something like aggregation of the lipopolysaccharide membranes, and like colonies are formed and large number of lipopolysaccharide membrane are excreted out from the cells, and they have a lot of proteins, extra cellular DNA, RNA, miRNA, and a lot of molecules are present. So, the aggregate of all is like a form biofilms which control the bacteria to survive for a long time. So, persistence of biofilm is basically the protecting the bacteria not to kill by various antibiotics or drug. So, interspecies communications it allow the bacteria to sense and to respond. For example, just like a chemotactic, phototactic bacteria. So, there are some signals which produce, they can sense where the light is there. So, they should go to the lights or they should come away from the light, or there are certain flagellar movement also, some tactic signals are produced. They can sense the signals, how where to go and how to swim and how to move in the water. So, these signals are controlled, communicated by the various quorum sensing mechanism. Antibiotic resistance and survival, again quorum sensing mediated the biofilm, which cooperative behavior can increase the tolerance of against antibiotics, various drugs, and various environmental stress. So, this is also controlled by the quorum sensing signals or quorum sensing molecule. Then therapeutic targets, again we can disturb these therapeutic targets of quorum sensing via autoinducers, analogs, and probiotics, and that's how we can control the infection, having the expression of these quorum sensing molecule. Now, I think this is last slide of this lectures, where we can summarize the talk is in the way that whole quorum sensing mechanism is basically important to understand, right, in different microorganisms, different class of microorganisms, in order to look into the connection between these quorum sensing molecule, how they connect, how they associate it, and what are the cellular and metabolic processes taking place so that they can produce certain molecules, signal molecules, they can communicate each other, they can cross-talk each other, and after cross-talking each other, they can say, okay, this is right, this is wrong for the bacteria. So, basically the quorum sensing mechanism is sort of the defense system, or it's sort of a offensive system also, we can say in both the way. It can also enhance the defense by certain means, by virulence factors. They can act as offensive. They can also cause more pathogenic, more infectious, more contagious, both in case of like a bacterial populations, some parasites, fungus, algae, and then they can have also interacted between the not only among the interspecies, but interkingdom, intergroup also they can connect. They can have from two different families, bacteria can mutually understand each other's, and they can try to communicate each other's. Now, it is the sort of understanding between two different individuals, two different bacteria, how they communicate. Sometimes, in humans, as human being also, we all are human beings. So, sometimes when we meet two individuals, each other, then either we can have good understanding with each other, or we don't have any good understanding. So, we call them as we don't have a good communication, we don't have a good understanding each other, so we cannot be a good friend. So, if you want to be good friend, we can live together, we can sit together, we can eat together, and we can like spend life together if you have a good friend. But if we don't have if you cannot communicate each other, if you not have a good understanding, rather we have a misunderstanding each other, then we cannot be a good friend. So, similarly in quorum sensing molecule, they provide either way, the two communicate two different individuals in a way that they can understand each other for their mutualism, symbiosis, or parasitism. So, just like human, I have given example, we can be a friend and we cannot be a friend, or we can be even enemy. So, similarly here The bacteria may be enemy for others, it can be friend for others, or it can be have a mutual understanding of each other. So this is all about the quorum sensing mechanism. I think you understand the quorum sensing mechanism, and this is some books you can go through, some research papers and some book. It is microbiology, McGraw-Hill editions, and then we can have a, this is the good review article, you can go through it, 2019 published by Mukherjee, and this is Nature Review Microbial. Having this understanding, I'm sure you can understand the smartness of the bacteria, smartness of the microbial world, how they can govern, they can control the whole environment, whole world. With this, I thank you very much.